All right, party people, we are back at it with some mitochondrial disorders. Let's get it. If I was to give you a 3-year-old boy with hypotonia, lactic acidosis, optic atrophy, some neurosensory hearing loss, and seizures, and I told you that he had a sister with hypotonia as well as myoclonus, which of the following is most likely characteristic of this patient's disorder? 3, 2, 1, boom. There it is. Let's get it. First and foremost, USMLE wants you to be able to diagnose mitochondrial disorders. The classic tetrad that you need to know is hypotonia, lactic acidosis, an eye problem, as well as an ear problem. This constellation points to mitochondrial disorders. Now, hypotonia and lactic acid makes sense because muscle tissue has high mitochondrial content, as does the eye, as well as the ear. So even if you're not sure of the exact diagnosis, if a vignette has these features, you should be thinking mitochondrial disorder. Now, the image shown here shows ragged red fibers. You don't need to know histopathology to a crazy degree, but for USMLE, you do have to be able to recognize these things. So in reference, the ragged red fibers are clumps of diseased mitochondria within the sarcolemma of the muscle fibers. That's what's shown here in the red. This finding is classically seen in Murph syndrome, that is myoclonic epilepsy with ragged red fibers. But again, USMLE doesn't care if you know Murph or Leber hereditary optic neuropathy by name. They just want you to be able to recognize a mitochondrial disease when it is presented to you. Now, in this case, with the 3-year-old boy, and he is severely affected, more severely than his sister. This is key. So what is the reason that he is more affected as compared to his sister? And the answer for that is the heteroplasmy. This is high yield that you need to know, and it's especially linked with mitochondrial disorders. To unpackage this, let's just get into exactly what is taking place here. Mitochondrial DNA follows maternal inheritance, passed only from mother to child. Males do not transmit mitochondrial diseases. So if they give you a pedigree chart, you'll see affected individuals only through the maternal line. This is unlike X-linked disorders because females can also be affected here. Why is it maternal only? That's basically because at fertilization, the ovum contributes mitochondria, not the sperm. So each cell has hundreds of thousands of mitochondrial DNA copies. When oocytes are produced, each one receives a different mix of these mitochondrial genomes. If a mother has diseased mitochondria, each child may inherit different proportions of the mutated as compared to the normal mitochondrial aspect. This is what is known as heteroplasmy. Variable severity of disease depending upon the proportion of maternal mitochondrial DNA inherited. So in this case, the 3-year-old boy most likely inherited a greater proportion, a greater fraction of diseased mitochondria than his sister. This led to more severe presentation. Now let's get into imprinting. Now imprinting refers to gene silencing depending on which parent the gene came from. You inherit 2 alleles of a gene, one from each parent, but one allele is silenced. There are 2 types of silencing that can take place. You can have maternal or paternal imprinting. So in the case of maternal imprinting, mom's allele is silenced. In the case of paternal imprinting, dad's allele is silenced. Now, why this happens, who the hell knows? But it does, and we have to know it. So examples are Prader-Willi syndrome. This is maternal imprinting. So mom's gene was silenced and dad's gene is deleted, all right? In the case of Engelmann syndrome, this is paternal imprinting. Dad's gene is silenced and mom's gene is deleted. Moving on to incomplete penetrance. Penetrance is the probability that a genotype actually shows up as a phenotype. To understand what we mean by genotype and phenotype, genotype is the genes, your genes, what is actually coded, and phenotype is what do we see in the person physically? How is it actually being expressed? Do you have genotypically coded blue eyes, and then do you actually have blue eyes? Because you might have a DNA sequence that codes for blue eyes, but in fact you have green eyes. So if you have blue eyes genotypically, but phenotypically green eyes are presented, this is how you make the distinction between genotype and phenotype. Genotype has to do with what's written in your genes, phenotype what shows up in reality. So if a disease is completely penetrant, everyone with the mutation shows symptoms. Examples of this are NF1. Incomplete penetrance means some people with the gene don't express the disease. For example, BRCA1, BRCA1 and 2. This can skip generations. Next up, variable expressivity. This refers to the variation in presentation despite the same mutation in fully penetrant conditions. So example of NF1 again. One person has neurofibromas, cafe au lait spots, as well as pheochromocytoma. Another one only has axillary freckling. They have the same mutation, but different expression. Everyone still has symptoms, just not the same constellation. As far as pleiotropy, This is where the gene causes multiple unpredictable phenotypes. Classic examples is Li-for-Many. Now, in Li-for-Many, this is TP53 mutation. One family member gets pancreatic cancer, another one gets colon cancer, the third one gets leukemia. They all have TP53 mutation, but the outcomes widely vary and it's very unpredictable. So what's the difference between pleiotropy and variable expressivity? So variable expressivity is complete penetrance. It's a predictable spectrum. An example that can be given is neurofibromatosis type 1. As far as pleiotropy, you have variable penetrance, you have unpredictable types of presentation, such as various types of cancers, and an example is Li-for-Many. This is the TP53 mutation. Just to spin the block and recap all this, mitochondrial diseases, you will see hypotonia, lactic acidosis, ear and eye problems. As far as ragged red fibers, these are diseased mitochondria clumped in the sarcolemma. For heteroplasmy, this is different disease severity due to differing proportions of mutated mitochondria. Also note that if you see a child with hypotonia and eye symptoms, with or without ear and with or without lactic acidosis and ragged red fibers, that's enough to suspect mitochondrial disease, even though not all of the features are present. So they can give you any combination of those, and you're going to have to be able to recognize that this is mitochondrial in nature. All right? That's it for this one. Go crush it.